Hello everyone, good morning all of you. This is Dr. Sudarshan Sambhajirao Pedge, Assistant Professor, Department of Zoology, Kailasa Rasika Mahavidyalaya Devani, your zoology companion, and welcome to my lecture. In the last lecture we also completed the introductory part of the metabolism of the protein. In that deamination as well as the amination process we have completed, or these are the transamination we also completed. So now today we will start next topic of our syllabus, that is the Ornithine cycle as well as Krebs cycle as well as the Urea cycle. Question may arise, what is the meaning of the Ornithine cycle as well as the Urea cycle? First of all, what is the meaning of Ornithine cycle? The conversion of ammonia to urea occurs by the Ornithine cycle. Okay, Ornithine cycle. Okay, what is meant by the Ornithine cycle? That the conversion of ammonia to urea occurs by the ornithine cycle, and it is also known as the Krebs cycle. Okay, as well as this cycle is also called the Urea cycle. Okay, this cycle is proposed, or these cycles who worked on it, that is the Hans Krebs and Kurt Henslevet, in the 1932 they have proposed, or they have worked on it. Okay, so this is a highly efficient mechanism in most of the terrestrial animals which remove the highly toxic ammonia in the form of urea. This cycle starts with the amino acid ornithine and hence called the ornithine cycle. Okay, as a result of deamination of amino acid and keto acid and ammonia are formed in the liver as well as in the kidneys, and this ammonia is highly toxic and should be removed rapidly from the body. One way of removal is by the formation of urea in the liver. Ammonia is converted into urea and keto acid oxidized into yield energy. Okay, that is the one particular thematic presentation. Okay, so you have seen here the cycles occurs generally in mitochondria and cytoplasm of the cell of liver, and it is also found out that the enzyme arginase, which is required in the final step of formation of urea, is present only in the liver and absent in all the other tissues. Okay, so this cycle generally occur in the only in the liver of mitochondria. Okay, mitochondria of liver and cytoplasm of the cells of liver. Okay, the following some reactions we have seen here, that is the various reaction in the process of urea formation. First of all, first reaction is the formation of carbamyl phosphate. In the formation of carbamyl phosphate, ammonia formed from deamination and carbon dioxide derived from decarboxylation combine in the presence of two molecules of ATP and H2O to form carbamyl phosphate. Okay, so in that, synthesized in the presence of the dicarboxylic acid, namely N-acetyl glutamic acid, which acts as a cofactor, and magnesium as well as the Mg plus ion. The reaction occurs in two steps. Okay, so we have seen here, that is the two picture is here in the reaction of this first formation of carbamyl phosphate. So you have seen here In the first step, CO2 is activated by one molecule of ATP in the presence of magnesium ion and N-acetyl glutamic acid. Okay, and in the next, the activated CO2 unites with the ammonia in the presence of another molecule of ATP to form a carbamyl phosphate, and this step is catalyzed by the enzyme carbamyl phosphate synthetase and cofactor N-acetyl acid and magnesium ion. Okay, you have seen here the carbamyl phosphate, it will be formed with the help of the magnesium ion and the as well as the uh, synthetase enzyme as a cofactor. Okay, so that is the first step of a formation of carbamyl phosphate. Another next step of this ornithine cycle, that is the second step, that is the formation of citrulline from ornithine. In the formation of citrulline from ornithine, the actual ornithine cycle starts only after the formation of carbamyl phosphate. In that, the carbamyl phosphate reacts with the S amino acids of ornithine to form a citrulline in the presence of ornithine carbamyl transferase. Okay, only with the help of ornithyl carbamyl transferase it will be formed, or it will be the transferase, it is the one particular enzyme, that enzymes it will be the form, that is the ornithine arginosuccinic acid. Okay, so after that you have seen here the next third reaction, formation of arginosuccinic acid. In the formation of arginosuccinic acid, citrulline then undergoes condensation with the amino acid as amino group. Okay, you have seen here the citrulline undergoes condensation with the amino group of aspartic acid to form arginosuccinic acid, and this reaction require ATP, magnesium ion and the enzyme arginosuccinate synthetase, and this reaction is a reversible reaction. Okay, you have seen here the third reaction is the reversible reaction. Okay, and in that reaction it requires ATP and magnesium ion and the enzyme arginosuccinate synthetase. Okay, and it will be the form the arginosuccinic acid, formation of arginosuccinic acid. Okay, and next reaction, that is the after the formation of arginosuccinic acid, the next fourth reaction, formation of arginine and fumaric acid. Arginosuccinic acid is then cleaved by the enzyme arginosuccinase to arginine and fumaric acid. Okay, it is one particular simple reaction you have seen here. In that arginosuccinic acid is it will be the cleave by the enzyme of arginosuccinase. Okay, and it will be form the arginine and fumaric acid. Okay, and last reaction you have seen in the ornithine cycle, that is the formation of urea. Finally, arginine is hydrolyzed by the well-known enzyme arginase into urea and ornithine, which completes the cycle. Ornithine is now ready to accept another molecule of carbamyl phosphate. Okay, ornithine is ready to accept another molecule of carbamyl phosphate, and the cycle gets repeated. Okay, you have seen here first of all citrulline, the formation of citrulline from the ornithine, the actual first second reaction. You have first of all the first reaction in that formation of carbamyl phosphate, after that the formation of citrulline from the ornithine. Okay, you have seen after that third reaction is the formation of arginosuccinic acid after third reaction. Fourth reaction is the formation of arginine and fumaric acid. In that arginine and fumaric acid, after the fumaric and arginine, the formation of urea is there. Okay. Finally, the arginine is the hydrolyzed by the well-known enzyme arginase into urea and ornithine. At that time, urea it will be formed, as well as ornithine is also formed. Okay. Again, ornithine it will be react or accept another molecule of carbamyl phosphate, and the cycle gets repeated. You have seen here. Okay. After the arginine reaction, urea it will be the form, as well as the ornithine it will be formed, but urea is expelled outside the body, and ornithine again react with the next carbamyl phosphate molecule, again repeated this reaction. Okay, so this is the ornithine, urea, as well as Krebs cycle. Okay, so now today we have seen here the ornithine, Krebs cycles, as well as the urea cycle. Okay, so thank you very much. Thank you.